Hi, my name is Shadie Nimri. I'm an application scientist at PhyTip, and today I'll be discussing the role of multi-platform PhyTip columns in cancer antibody research. So since the therapeutic use of antibodies continues to grow for cancer, since Kohler and Milstein's work on the hybridomer technology, the need for high-throughput antibody purification has become a staple in many of the areas of antibody discovery and the development process. Whether it's through early work in the mechanism of action determination, whether it's purifying a large number of samples in high-throughput screening, or higher level characterization studies, automated small throughput purification can enable more research, shorten workflows, and standardize results. So what options are there for automating small-scale protein purification? The two main avenues are by using filter plates or magnetic beads. Filter plates are the most cost-effective option and probably the most widely used. However, the single pass-through can lead to sample loss and possible contamination. The need for filtration also requires a vacuum, which is a barrier for automation and can also lead to clogging. Magnetic beads are the second most popular option, and there are a lot of options for automating this process. Also, there is no filtration needed, so there is no clogging, and it's easier to automate. However, it still does require specific instrumentation, which can become a large capital investment. And there is inefficiencies in binding and washing. And some of the inefficiencies in binding can require more magnetic beads, which can drive up costs and can lead to smaller concentrations. Also, inefficiencies in washing can lead to carryover in contaminants. So today I'll be focusing on PhyTip columns, which are a pipette tip-based chromatography column with resin trapped between two inert screens. There are platform-specific tips, which are robotic instrument tips modified with this resin. There are 200 and 1000 microliter column volume options. And PhyTip provides resin volumes packed between 5 microliters and 320 microliters. And these tips come standardly packed with a variety of chromatography resins. PhyTip also offers a custom packing service, so essentially any commercially available resin can be packed. So how do PhyTip columns work? PhyTip columns cycle sample back and forth through the resin bed, which we have dubbed dual flow chromatography, and follows the traditional steps of chromatography in terms of capturing, washing, and elution. Some of the advantages of dual flow chromatography are the process control. You can calculate the residence time by controlling the number of cycles, flow rates, and mix volumes. So it's easy to translate traditional chromatography to dual flow chromatography and scales well with larger columns. Also, you can control and maximize the residence time to ensure that all of your sample has bound and eluted off the column. PhyTip columns are manufactured and compatible with most of the liquid handlers on the market, particularly Hamilton, PerkinElmer, Beckman, and TCAM. PhyTip also manufactures a small to mid throughput instrument called the MEA2, which is compatible with all the PhyTip columns. PhyTip columns are manufactured from the raw tips of the robotic instruments. So if they're compatible with the tips that are processed traditionally on your instrument, they will be compatible with the PhyTip columns. One of the two major advantages of PhyTip columns specifically is that there is no capital investment. The columns are ready to use out of the box and do not require an adapter, so they do not lock the robotic instrument. The columns come with templated robotic scripts, which can be used to customized workflows, such as the one for Hamilton, displayed here. And there's no column prep. And the columns can be used 1 to 96 at a time and can be run parallel with multiple resins for any testing. The second major advantage is the concentrating effect of the packed column, which is useful for processing small volumes. Here we have 10 microliters of sample being pipetted back and forth through the column. And as you can see, the 10 microliters is fully aspirated into the column, and all of the sample comes into full contact with the resin bed, and the full volume is expelled. So there's virtually no dead volume, and this leads to higher concentrated samples as compared to other platforms. And that result is also highlighted here. An experiment performed courtesy of Vertex Pharmaceuticals. In this experiment, his tagged proteins, kinases, were compared against spin columns, magnetic beads, and PhyTip columns. With the same amount of sample using the platform's suggested protocols, the eluted sample is concentrated enough to be analyzed on STS page from PhyTip columns, but is hardly visible from spin columns or magnetic beads. That same concentrating effect is also highlighted here. This is human IgG processed through pure spin columns and through 20 microliter and 5 microliter resin bed. Here we have the standard 0.5 mg/ml human IgG with alternating flow-throughs and elutions analyzed on this gel. And as you can see, the 20 microliter resin bed elutes sample concentrated enough to be analyzed, and the 5 microliter resin bed can be eluted in even smaller volume and leads to even higher concentrations. So the most obvious application for high throughput small scale purification is expression screening. You have to test multiple expression systems in parallel, so you have to purify a lot of samples. Must test for solubility and stability, test many different conditions, and test for protein function and characterization. So you have to supply protein that is concentrated and pure enough that you can provide confident primary assay results. We have a few examples of customers using PhyTip columns for expression screening. Here is an example from a group at Roche doing X-ray crystallography screens and purifying multiple constructs in multiple expression systems using PhyTip columns. And they're consistently able to get enough product to make decisions for each construct on an SDS page in each expression system. The same group then took one construct and optimized for binding efficiency and protein stability. They tested an array of 96 different buffer conditions using a hummingbird robot, and then took the 2 highest yielding buffers and tested for stability overnight, and found that tris hydrochloride, pH 8.5, in a specific detergent was found to be the most high yielding and stable buffer condition. This is another example from a user at Cambridge Antibody Technology doing cell-based screening on single-chain variable fragments. They found that the product purified by PhyTip columns was able to provide a higher resolution inhibition curve as opposed to their standard pyroplasmic extraction protocol. And when scaling up the product using a larger scale IMAP column, they found that the IC50s between the smaller scale product versus the larger scale product was consistent between each construct. PhyTip columns also provide a perfect platform for automating long immunoprecipitation protocols. Immunoprecipitation itself is a critical tool in investigating post-translation modifications on antibody-based drugs, as well as studying drug-target interactions. PhyTip columns provide a perfect platform because target-specific antibody can be pre-conjugated and packed into the columns. And since the columns are stored in a buffer containing glycerol, the antibody is stable for a very long time. And having the pre-conjugated antibody cuts the processing time in half while automating the rest of the protocol. So here's an example from a group at Genentech who was purifying KGG modified peptides in a complex mixture using immunoprecipitation. And going from a manual procedure of multiple binding and washing steps using a centrifuge, they used pre-conjugated Pro Plus PhyTip columns with antibody and used an automated procedure on the MEA robots. So the process consisted of lysing their target and using the PhyTip columns to bind KGG specific peptides and then analyzing on LCMS. What they found that automating the procedure, one, gave more consistent, but also more specific KGG peptides as opposed to non-KGG peptides, which increased the LCMS resolution. So scaled down purification is also a very important step in process development. So why would you want to scale down your purifications in process development? Well, you can screen a wider range of protein conditions and purify lots of samples, require less material, which saves time and money, and you get simulated performance of larger columns. So scale down purification is useful in upstream process development as well as in downstream. Upstream, the goals are to determine clone selection, develop cell lines, and to analyze key quality attributes. And downstream, you want to predict the performance of larger scale columns and perform simulated gradients to optimize those columns. Here is an example of scale down purifications done from a group at Merck. And first, they optimized the binding efficiency of the columns to allow very little flow through, sample left in the flow through, and to maximize yields with yields of up to 6.5 milligrams per PhyTip column. Then they did a study to compare large-scale product versus small-scale product and studying different quality attributes, in particular the charge variance and looking at aggregates. And in both purification scenarios, the quality attributes were consistent throughout. So here, the PhyTip columns were used both on a TCAN in blue and they were used on the Biotage MEA in red. PhyTip columns have also been used to automate plasmid purification and provide completely automated solutions for high throughput mini prep, as well as larger scale Maxipreps. So some of the key challenges in automating the state-of-the-art technology is that the cell lysate, the E. coli cell lysate, requires to be removed by centrifugation or filtration. Not only is this not an automated step, but filtration can also lead to clogging as well as compromised yields or purity. So to overcome this challenge, the mini prep PhyTip column has been designed to be a loose packed silica-based column, which is able to directly bind DNA in the presence of the cell lysate with no need for centrifugation or filtration. And this allows the entire process to be automated, including the resuspension, lysis, and precipitation. These PhyTip columns provide, using high copy plasmids, yield plasmids of over 10 micrograms, which are also transfection grade. This is a comparison study done by Genentech, comparing the manual standard mini prep process to the automated PhyTip process. What they found was that both processes provide comparable yield as well as comparable transfection efficiency in multiple transfection systems. A different approach has also been taken for automating the larger scale Maxiprep process. The Maxiprep PhyTip columns are 20 ml columns as opposed to our 1 ml columns, and they utilize a unique liquid handler called the PhyPrep. Designed by Biotage, the PhyPrep is a 2-channel liquid handler, and is specifically designed for automating Maxipreps. And it's a press-and-go system that is able to produce transfection-grade, endo-free plasmid. Maxiprep system with high-copy plasmids consistently yields plasmid over 1 milligram that is endotoxin-free. In summary, PhyTip columns are a pipette tip-based chromatography column that are compatible on most liquid handlers. They enable the high throughput purification of antibodies in many of the areas of drug discovery and development. The PhyTip columns yield highly concentrated samples, and they also provide a level of control and flexibility compared to other types of automation. And the packed bed chromatography provides results that is both reproducible and scalable. Since I have a little bit of time left over, I did want to highlight some new and developing applications for PhyTip columns. Recently, Capture Select resin has become a standard PhyTip option for the use of both antibody chain and fragment purification, as well as AAV purification using Capture Select AAVX resin. And this AAV purification process is being optimized with collaborations with Spark as well as the UMass AAV manufacturing facility. PhyTip columns have also recently been used to completely automate the mass spec sample prep workflow, using both desalting PhyTip columns as well as trypsin digest columns. The trypsin digest columns are packed with immobilized trypsin manufactured by Promega. And with the digest done on the column, the entire process can be automated up until the dry down. So the process is shortened by nearly tenfold.
Thank you for your time, and please contact us if you have any questions.